discovery. We embrace the unwavering belief that with passion, determination, and a spirit of resilience, nothing is truly out of reach.
I love biosensors. I love talking about them, I love thinking about them, and most importantly, I love working with them. However, I know from experience that most of us in this room have no idea what a biosensor is. My goal for today is to have all of us leaving here a little bit more informed and hopefully all equally as excited about this amazing technology. So let's break it down. If I asked you to call to mind biology as a subject, you might flash back to the last time you sat in a science classroom. Maybe you'd call to mind imagery of cells or DNA or proteins. If I asked you to call to mind sensors as a broad class, that imagery might become a little bit more fuzzy, though I promise your experience with them is just as tangible. Maybe it's the last time you had your vitals taken in a hospital setting, maybe an at-home test that gave you some good or not so good news, depending on the test, or the check engine light that we've all had on in our car for probably just a little bit too long. Sensors are all around us, and they enable the modern world we live in. So naturally, when I tell people that I work with biosensors, the next question that I usually get asked is, so you use DNA to make sensors? To which I give an enthusiastic yes, because it really is that simple. But we'll come back to that. First, let's start with this number, $750 billion. $750 billion was wasted on unnecessary healthcare and mistreatment services in 2009. $750 billion. If you're anything like me, you have no way of comprehending a number that big, so I broke it down. For $750 billion, I can take every high school senior at Laguna Blanca, and I can send you to a 4-year university of your choosing with full tuition, room, and board all paid for. Who's game? In fact, for $750 billion, I can do this for every single high school senior in America, all 3.9 million of them. This is a huge sum of money, and yet every year we waste it. So the question then becomes, how do we mitigate these losses? Well, in the past few years, we've all become intimately familiar with how important sensitive and accurate testing can be for human health. During the COVID-19 pandemic, at-home tests enabled individuals to be informed about their health, prevent viral spread, and reduce strain on already overwhelmed healthcare systems. These sensors work by using a protein that's been labeled, called an antibody, to engage whether there's a virus present or not. If the virus is not present, the antibody won't be able to bind it, and you'll get a negative test. If the virus is present, you'll get a positive result after the antibody binds, indicated by 2 red lines. This class of sensors is amazing for if a simple yes or no is all that you need. However, some instances, I need a quantified result, or maybe even results in real time. What then? If you have or know anyone who has diabetes, then you know that infrequent sampling can lead to missed highs and lows in blood sugars. Luckily, biosensors have answered this need as well. Continuous glucose monitors enable real-time glucose measurements, which give patients the information they need to treat accordingly. Continuous glucose monitors use the enzyme glucose oxidase to oxidize glucose into gluconic acid. The byproduct of this reaction is oxygen, and we can quantify this electrochemically and then relate that oxygen concentration back to the glucose levels in their bodies. This kind of technology has been around for about 25 years and works great for most patients. So why then aren't we familiar with biosensors for more than just glucose? Well, if you go back again to that biology classroom I sat you in, enzymes like glucose oxidase are proteins that are made up of 20 unique amino acids, and they're arranged in hundreds of thousands of possible combinations. These molecules are incredibly complex, and as we stand technologically, we just can't engineer them yet, unless Mother Nature already has an interest in them, like in this case. So how can we simplify this problem so that we can make a bioreceptor in a test tube? Well, I kind of spoiled things already by telling you that I make my sensors with DNA. In fact, in the Plaxco group at UCSB, we've helped develop an entire class of sensors called electrochemical aptamer-based sensors, or EABs. These sensors use DNA as their biorecognition element of choice. Why do we prefer DNA for our sensors? Well, DNA uses just 4 nucleic acids, those A's and C's and G's and T's, and the short strands of single-stranded DNA that I work with, called aptamers, use typically about 30 to 90 nucleic acids. As monkeys in lab coats, that becomes much easier for us to synthetically engineer and modify. In addition to being able to be synthetically engineered, these aptamers are also highly generalizable, meaning that I can make them for any target I have an interest in, whether that's a drug or a biomarker, maybe even a protein. Similarly, they enable non-reactive measurements. So unlike the glucose meter, where you're relying on the reactivity of glucose, aptamers are completely non-reactive with their targets, meaning they unobtrusively bind and unbind whatever they're detecting. How do we make them do this? Well, we intelligently design them so that when they are in the presence of their target, they undergo what's called a conformational change. Essentially, they fold or change shape when their target of interest is introduced. We engineer them so that they also do this reversibly, so that as concentrations change, the aptamer can unbind and return to its unbound state. We exploit this shape change by adhering one end of the aptamer to an electrode and the other to a redox reporter, which is a label. The redox reporter's whole job is to transfer electrons to the electrode surface when we interrogate it electrochemically. When the aptamer undergoes its conformational change, this rate of electron transfer changes. We can then relate this electron transfer rate to concentration, and boom, easy as pie, you've got a biosensor. What's really cool about these sensors as well is that they're less than 1 millimeter in diameter. This means we can fit them inside a catheter and easily take measurements not only in a vein in your blood, but also in the minimally invasive subcutaneous space right below your skin. Similarly, for research purposes, if we're curious about how a particular molecule distributes throughout your body, we can take measurements in tissues like muscle or even the brain. So how is this kind of measurement relevant to you? Well, if you've ever had a smartwatch, you're already intimately familiar with how real-time biometrics like heart rate can inform things like a workout, your sleep, or even your mental health. My heart rate, for example, is very high right now. However, you may also know that this metric is going to be different from the person sitting next to you based on your unique physiology and experience. This highlights exactly why individualized healthcare, a sensor that quantifies your unique metabolism and distribution, can have a huge impact on your health. Furthermore, the clinical applications of EAB sensors becomes readily apparent. In a hospital setting, lab results for both diagnosis and treatment are typically collected at most once daily. This leaves clinicians only able to blindly or reactively dose or treat based on out-of-date metrics like BMI or single-point lab samples with hours to days of lag time. This all leads to the wasted time, tests, and suffering that compounds to that $750 billion we discussed earlier. The stakes become even higher for drugs with very narrow therapeutic ranges like chemotherapeutics. Not enough, and you don't treat the cancer. Too much, and you face increased risk for adverse side effects. Take, for example, the bronchodilator Theophylline. Theophylline is administered orally to patients with severe trouble breathing for whom inhalers aren't compatible. However, it has an extremely narrow therapeutic range and needs to be dosed accordingly to avoid giving patients who are already ill increased risk for seizures, nausea, or even cardiac arrest. Here in this graph, you'll see what is clinically traditionally used to inform diagnosis. It's 4 actual measured data points over a 6-hour period, and then this black line is extrapolated between those data points. 4 measurements over a 6-hour period. In this graph, you'll see an actual measurement we collected for theophylline in plasma using an EAB. The difference being that each dot you see on this graph is an actual measurement. No need to extrapolate or guess between the points. This is a relatively simple example. However, I encourage you to think of which option you would prefer a clinician use to inform your treatment, given the stakes are so high. But how does this affect you in day-to-day life? Well, what if that same smartwatch we talked about earlier could quantify information you would normally only get maybe once a year from a blood panel, but on an hours or daily basis? Maybe we quantify your lactate levels after or during a particularly hard workout, your tryptophan levels as a precursor to serotonin after a big meal, or maybe even your serotonin levels directly as an indicator of mood or potential sleep. These are all targets that the field is currently working on or has sensors for. This amazing future is what has me so excited and why I think biosensors will do amazing things for all of our lives. All of these would be super impactful. Soon, biosensors will be just as ubiquitous as our smartwatches, and we'll all hopefully live a little bit better and healthier because of it. Thank you.